Salutations. My name is Liliana Nemechek, and I will be presenting notes on the ground-dwelling arthropods of Pitcairn Island. Islands make up just 7% of Earth's landmass, yet they support approximately 20% of the planet's biodiversity. This extraordinary richness is due to their geographic isolation, small habitat sizes, and unique disturbance histories. These same factors also make island ecosystems, and the species they support, particularly vulnerable to environmental and human-induced change. Nearly half the species listed as threatened by the IUCN are island endemics, and nearly 70% of known extinctions have occurred on islands. Located in the South Pacific between French Polynesia and Rapa Nui, Pitcairn is one of the 4 islands in the Pitcairn archipelago. Home to about 50 people, it's the only volcanic and inhabited island from the group. Pitcairn Island was settled in 1790 by mutineers from the HMS Bounty and their Tahitian companions. Like the Polynesians before them, these settlers brought livestock and crops, not just for survival, but to perpetuate the Polynesian way of life. Such introductions, while essential for settlement, are known to have profound impact on fragile island ecosystems. Today, Pitcairn's arthropod community remains largely undocumented, which makes it difficult to assess how environmental changes and human activity might affect them. This study provides a first look at the ground-dwelling arthropods on Pitcairn Island, setting the stage for future research on the island's arthropod communities and any species that might require protective management. Over 2 days in late June of 2024, 3 researchers collected arthropods in 2 coastal and 2 inland locations. They focused their efforts on pockets of native vegetation, as these locations were most likely to support native arthropod communities. My job was to sort and organize the specimens collected during this effort into operational taxonomic units, or OTUs. An OTU is a group of organisms determined to be morphologically similar. However, I will now refer to OTU as species. As I am presenting preliminary results, most specimens have been identified to the order or family level thus far. However, we did identify several to genus and species level. As a result, we identified 88 species. Of this island assemblage, spiders were the most diverse group, accounting for 23 species. The second most diverse was Hymenoptera, with 7 morphospecies. We also documented a new island record for Pitcairn, the weevil species Oxydema longula. This beetle is also known to neighboring Henderson Island. However, until now, it was not known to occur on Pitcairn. Perhaps our most exciting finding thus far is that we have confirmed a new species of springtail, or columbella. At about 1.5 millimeters, about half the length of a grain of rice, this tiny animal belongs to the family Neanuridae and is of the genus Australonura. As this is a new species, we believe it also represents a species endemic to the Pitcairn archipelago. This means it probably occurs in this island chain and nowhere else on the planet. As our research progresses, the next steps will be taken to more fully address some unanswered questions. First, we need to complete the identification of specimens. This will involve both additional research of selected specimens and sending some specimens to taxonomic specialists for confirmation of identifications, as well as formal description of new species. This work will enable us to more robustly identify which species are non-native and which are endemic. For endemic species, we will want to know whether they are endemic to the Pitcairn group or to Greater Polynesia. Species endemic to Greater Polynesia could be canoe bugs. So far, there are 3 potential canoe bugs known from the South Pacific. If we discover additional examples on Pitcairn, it would provide additional support for the canoe bug hypothesis. In time, we may be able to apply a molecular clock analysis to trace when and how these species dispersed across the Pacific. We've started to formally describe the new springtail species. According to Dr. Adrian Smallis, a columbella systematist at the University of Wrocław in Poland, our new species is most closely related to a species first described in Australia and later found on the Solomon Islands. To best describe this species, Dr. Smallis, Dr. Wynne, and other collaborators will compare the Pitcairn species to the Australian species using DNA barcoding analyses. And finally, once the springtail is formally described, protecting it and other endemic species should follow. One of the greatest threats to both native plants and endemic arthropods on Pitcairn is the rose apple, Syzygium jambos. Introduced over 2 centuries ago for firewood, this tree species is highly invasive. This highly invasive species forms dense monocultures that suppress native vegetation and impede the growth of native plant species. With effective management of the rose apple and other invasive species, Pitcairn's native habitats can be enhanced, and in some cases, restored. Removal and control of non-native plant species will help safeguard the island's endemic invertebrates. We would like to give a big thank you to the NAU Interns to Scholars program for providing funding for our research. As well, thank you to Darwin 200 and Exploring by the Seat of Your Pants for providing funding for the fieldwork. Thank you to Gary Bobker, Benny Lindberg, Mike Lupton-Christian, and Gerald Prahl for assisting in the field. Jutwin thanks the captain and crew of the 3-masted schooner, Oosterschelde. Thank you to the NAU Arthropod Imaging Lab, who contributed tremendously by providing imaging training and support. And finally, research was conducted under a permit granted through the Pitcairn Island Marine Protected Area Ordinance 2016. Thank you again to all of our contributors, and thank you for watching.